Hello students, today in this video session we are going to discuss about the remaining part of animal kingdom, that is phylum Porifera. Yes, in the chapter kingdom Animalia, that is what we call animal kingdom, in that phylum Porifera. This is the first phylum of invertebrates and in the animal kingdom, right? So Porifera, the meaning of this phylum is that is porous. The body of the members of this phylum, all members are showing the porous body, hence the name is Porifera, and also commonly known as sponges, commonly called as the members belongs to the phylum Porifera are also called the sponges, because the body is like a sponge, hence we called them as sponges, right? So Porifera, the porous body. So how? Let us see the structure of the one of the example we will take, that is cycon. The example, there are many example we can take. Cycon, also called scypha, okay, cycon, and spongilla, euspongia, yes, and there are many other examples you can take. Euspongia, bathing sponge. So euspongia is also referred as bathing sponge. What do you call? Bathing sponge. So this is also called bathing sponge. Bathing sponge, so which is used for bath because the endoskeleton, we will discuss it later. Okay, euspongia is one of the example you can take. And next, so euplectella, you also take the example of euplectella. It's also called Venus flower bucket, Venus flower basket. So like another terms are there to define this one. So that is the euplectella is also called Venus flower basket, right? And other examples are Hyalonema. Hyalonema is a glass sponge. Hyalonema, you take Hyalonema is called glass sponge. It's called glass sponge. It appears like a glassy. Okay, hence the name is glass sponge or Hyalonema. And euplectella is also known as Venus flower basket because which is the just like ornamental things which is used, the endoskeleton, how we will understand that. Okay, endoskeleton of this animal, that is euplectella, is used as a ornamental or gift items, like it is used. And the euspongia is also called bathing sponge because the sponge like it is, and it is used to wipe this tiles and floors. Even for bathing also the peoples are using. Yes, so cycon, another example, cycon and spongilla. See here, out of these examples when you observe, so spongilla is a freshwater form. It's a freshwater. Of course these all are, to take here, all are multicellular, and they are aquatic. All are aquatic. They are found in water, aquatic, and most of them are marine. And few are freshwater form. Few are freshwater. So one of the example what I have given here, spongilla is a freshwater form. Freshwater. So this is one is a freshwater, and remaining what we have taken here, the examples are the marine. Marine means they are found in ocean or sea. That's what salt water. They are found in salt water. and the spongilla is one which is found in the fresh water, like freshwater lakes and big water bodies which may be present. Yes, so all are exclusively aquatic. They are exclusively aquatic, right? So this is what the one of the important criteria we can say. The phylum Porifera have their own features, general features. Body is porous and they are all are multicellular primitive animals. These are multicellular, primitive multicellular animals we can say. Primitive means very what you call simple. Okay, so and organization, when you observe in the next point, the cellular level of organization we observed here. What we call cellular level of organization. Cellular level of organization we observed in this one. Cellular level, what it means? Cellular level means still tissue is not formed. Okay, when you observe the body structure, let us understand by this one the structure of the, and their body is as you know asymmetric. Even they are multicellular, they are showing the asymmetry. Asymmetrical body, they shows asymmetrical body. So you have to note the points, very important points related to this one. They are multicellular primitive animals, primitive multicellular animals. Next, they are all are aquatic. See here, all are aquatic and mostly marine. Few are freshwater forms. Few are freshwater forms, and all they shows see here, they shows that cellular level of organization. Cellular level of organization, very important. So and all are showing the most of them. Okay, asymmetrical body. Asymmetrical. There is no symmetry. Asymmetry we can say. Right, there is no bilateral or radial symmetry. They are not become two equal halves, as you know, right? So asymmetrical body are there. Let us understand the let us take the structure of cycon. So mostly they are usually they have no locomotion even. Okay, so primitive these are, and they are attached to the rocks or the substratum which is present in the water bodies, right? So they are usually, for example, this is a substratum you can consider, and so this is what the cycon structure what I am going to draw here. Yes, this is what the external view. So this is somewhat, this is the big opening here, and so body has porous structures like this. There are many pores. We can observe it as many pores which are there in the body wall. In the body wall, there are innumerable number, we can say infinite number of pores are present through which water enters into the central cavity, right? So water enters into the central cavity through this pores. In this way, the water enters here. So these pores are called, yes, ostia. That pores are called ostia. Yes. So ostia are the pores present on the body, and which are allowed to enter the water inside the cavity. That means inside the body. So that is internally there is a body cavity is there, right? So this is what the substratum, and this is the example of cycon you can take. So cycon, and that entered water which is there internally here. So when you observe the sectional view, so the internal cavity is there internally, the central cavity is there, but it is not referred as body cavity. You remember, already I have explained this one. Yes. Next, this see, the entered water then exit through this large pore, the large pore. So this pore is called osculum. What you call osculum, through which water is exit from the central cavity of that animal, right? So ostia are the pores, minute pores, they are innumerable number, and through which water enters inside the body. But the osculum is a little large, and usually one osculum they have, and usually here it is allowed to exit the water. water is exit here, right? So this is what the functions of ostia and osculum, you remember, right? And when you observe the sectional view, so the pores, let us try to understand by the sectional view of that diagram. See, so see here, so this is the body wall you can consider. So sectional view, when I am drawing here, so this is what the substratum here, this is the substratum, and it has many pores. You can see there are number of pores. You can show the pores here, right? These are the pores, right? These pores are called ostia, and this is the outer layer is called pinacoderm, and inner layer is called choanoderm. There are two layers, diploblastic. We can say these animals are diploblastic. They are diploblastic. We can add the point, diploblastic animals. They have two layers, outer pinacoderm, inner choanoderm. So here the choanoderm is made up of a specialized cells called collar cells or choanocytes. They are formed by choanocytes or collar cells. They are very specific in structures. They are like, this is the spherical structure with the collar shaped structure which we observed, and along with that there is the presence of flagella. So along this is the flagellum, and this is what the cell. Yes, this is the cell. This is like a structure called collar. Okay, in this type. And in pinacoderm, outer one is called outer layer is called pinacoderm, right? Pinacoderm shows that it is made up of a special kind of cells, flat cells. They are referred as pinacocytes. What do you call? Pinacocytes. Pinacocytes are flat cells. For example, like this. The cells are like this. They are flat cells. In this form, cells are there. Okay, only two layers which we observed here. Inner, the choanoderm. Inner one is called choanoderm, and outer layer is called pinacoderm. So choanoderm is made up of choanocytes, or also called collar cells, which are like this. So you can observe the cells, and the pinacoderm, which is the outer layer, and it is formed by the special cells, flat cells. They are called pinacocytes, right? So these are the two types of cells which we observed in these two layers, hence we refer it as diploblastic. And centrally, in the middle, so there is a what you call the jelly like substance, water and some collagen fibers, which is made up of collagen fibers, that is also called mesoglia, which is made up of usually water and collagen. Yes, this is what the thing which is present in the middle space. Okay, so it's not a middle layer. Okay, because there is no cells, there is substance, but that also shows some cells, some irregular shaped cells. So they are called amoeboid cells. What we call here, we can observe that amoeboid cells. They are referred as amoeboid cells which may secretes the collagen, and forms this mesoglia. Yes, mesoglia we called. Next, this is the central cavity. The central cavity is known as yes, this central cavity is called the central cavity, that is referred as spongiocele. Spongiocele, right? This is called the spongiocele. This is what the central cavity, but it is not body cavity, as I referred already. Spongiocele, okay, the cavity which is exists in the central, right? And as you know, these are the pores which are formed by the special cells here, elongated cells. These cells are called pore cells. Pore cells which forms the ostia, right? So this is about the body wall, and this is as well osculum, right? So when you enlarge this portion, if I enlarged here the portion of this much, so the inner layer, see, this is what the inner layer, and this is what just I am showing you. So this is the outer, right? These are the pinacocytes, right? Pinacocyte cells, and collar shaped cells you can show in this form. This is the quinocytes with flagella. There is a structure called flagella. So this is what the structure, and this is the cells, elongated cells, pore cells, and there may be amoeboid cells. Yes, these are the amoeboid cells, and there is a presence of spicules in this layer. There is a presence of spicules. They may be needle-like, or branched or unbranched needle-like, right? They may be existed here. Yes, in some sponges it may be made up of spongin fiber, or maybe calcium carbonate, or the spicules may be made up of calcium carbonate. There are spicules are of two types. Spicules, these are called the spicules of two types. One is made up of calcium carbonate, CaCO3, and another one is usually silica. Yes, silicic acid, silicates, we can commonly say it as silicates. Okay, so based on this content, spicules are of two types, maybe unbranched or branched, right? Needle-like, they are needle-like. And another is what you call spongin fibers, which may be this forms the what we call endoskeleton. So they have an endoskeleton, right? They have endoskeleton which gives the support. Okay, so there is a existence of endoskeleton in the mesoglia, right? In this way there is a presence of endoskeleton here. Yes, so this endoskeleton is formed by either calcium carbonate or silicic acid, silicic acid, right? And spongin fibers, right? So these are about the body structure, and the remaining digestion and other structures we will discuss in the next video. Thank you, students.